Hi everyone, we're back with video number 5 of 5 in lecture 31. Let's go. I showed you this before, this slide, when we talked about inductive signals as one of the two main initiators of cell differentiation, the other one being the cytoplasmic determinants we talked about in video number 4. So inductive signals come from other embryonic cells, and they cause changes in gene expression. And here's an example. We talked about this before, but just a quick recap. This little panel in the lower right here on the left shows two types of cells. One cell is the brown, shown in brown, and the second cell down below in green. And in this little schematic, the green cell is releasing signaling molecules which are picked up by the receptors in cell number 1. So the receptors were already there in the cell. So there was enough development in the brown cell previously to turn on the genes to produce these receptors. Likewise, in cell number 2, there were enough genes expressed to turn on the production of the signaling molecule. Once the signaling molecule binds to the receptor in cell number 1, that initiates some kind of signal transduction pathway which turns on other genes, and so therefore the green cell has signaled to the brown cell to do something. Turn on genes, turn off genes, etc. All right, let's look at an example. And that example is going to look at inductive signaling in C. elegans. What is C. elegans? C. elegans is a worm that is used in research quite a bit. It's a cool research vehicle because there are only 959 cells in the organism. So as a researcher, you can almost know what every cell does, and we can look specifically at development. So in C. elegans, eggs are laid through the vulva, which is shown here in this picture, and we're going to look at development of the vulva. And this is an example of inductive signaling. All right, so here's how it starts. We've got an anchor cell, and we've got these vulva progenitor cells. The anchor cell sends out a signaling molecule. It's EGF, or epidermal growth factor. So they're positioned somewhat like shown in the cartoon here, where the anchor cell is above the vulva progenitor cells, and the EGF ligand is sent out. The progenitor cell nearest the anchor cell receives the most ligand, so that cell is shown here in red or in pink. That turns on a signal transduction pathway to make this progenitor cell into a type 1 cell. So we started out that all the three progenitor cells shown here were the same. The one that receives the most EGF ligand, the EGF ligand binds to a receptor. There's a signal transduction that happens. The signal is transduced inside the cell, and certain genes are turned on or turned off to make this a type 1 cell. So it's the anchor cell that induces the development in one of the progenitor cells. Type 1 cell then, once it becomes type 1 cell, part of its programming is to send out signals to its neighbors. And the signals do a couple things. One, they prevent the neighbors from being another type 1 cell, and they tell the neighbor, go ahead and become a type 2 cell. And this is how development can start to spread outward from the signal originally sent by the anchor cell. So this is pretty detailed. Let me go through it first, and then you probably want to read all the words. But here's our little picture that we just talked about up here. All right, so the anchor cell sends out the ligand, and that induces changes in the progenitor cells down below. The progenitor, the red cell, type 1 cell, does lateral inhibition of cells next to it. So It starts out with the EGF ligand shown here, binding to the EGF receptor. So the progenitor cells have EGF receptors in them. Look at both the red cell and the blue cell. They have the EGF receptor cell, or receptors already in them. So that was part of earlier development. They also have another receptor called the Notch receptor. And look at both cells have the Notch receptor to start. So the EGF can bind to both EGF receptors, but because there's more of it around the cell that becomes a type 1 cell, that's the red cell, because there's more EGF receptor received by the type 1 cell, it's the one that converts into type 1. All right, it was a normal progenitor cell and converts into type 1. All right, so then what happens? Well, when the EGF ligand binds to the receptor, it starts a signal transduction pathway that goes through these steps. Like we said, we've seen this before, and amplifies the signal, and it actually ends up in the nucleus turning on certain genes that cause this cell to become a type 1 cell. Part of what being a type cell means is, one, it produces genes, or in this case probably turns off genes that inhibit production of the Notch receptor. So we get rid of the Notch receptor. The Notch receptor we want to use over in a type 2 cell. So we're going to turn off the Notch receptor in the type 1 cell. Secondly, we're going to produce a ligand that is going to bind to the Notch receptor. So we produce the Notch ligand here in the type 1 cell. It is released and picked up by the Notch receptor. So because we are producing in type 1 cell a Notch ligand, we want to get rid of that Notch receptor in the type 1 ligand, so we're not signaling ourselves. So that's why we knock down the Notch receptor. The Notch ligand is produced, and it binds the Notch receptor in this other cell, which leads to the signal transduction pathway in cell type 2, and that induces this cell, this progenitor cell, to become a type 2 cell. One of the things that being a type 2 cell means is it blocks the EGF receptor. So while we had the EGF receptor in the cell that becomes the type 2 cell, we turn that off and get rid of it. All right, so the EGF ligand is no longer going to be received or is no longer going to bind at all to the type 2 cell. And we turn on other genes that make this a type 2 cell. All right, so that's how inductive signals work. Pretty cool stuff. So please read through the details and understand this pretty well. Here's how it looks in the actual C. elegans worm. In our cartoon here, we have the type 1 cell, or in this case it shows two type 1 cells having received the EGF receptor, and then developing the type 2 cells next to it. And here are the actual pictures in C. elegans itself. So I said the anchor cell and the progenitor cells were organized somewhat like is shown in our cartoon, and you can see that here. And so the type 1 cells are marked, and the type 2 cells are marked, and the anchor cell is marked. So as you get more cell division, you end up with the production of the vulva, just like shown at the bottom of this slide. Finally, the EGF binding, the EGF receptor is a receptor tyrosine kinase. So do you remember those way back when we talked about signal transduction? The signaling molecule binds to the receptor tyrosine kinase. Two of those come together and they phosphorylate each other. They activate a signaling protein, and signal transduction happens as shown on the right. So you don't need to know all the details of this cartoon. Just know that it's a little review really of the fact that receptor tyrosine kinase is one of those key receptors, one of the 3 types of receptors we saw when we talked about signal transduction. That's it for lecture 31. Thanks for watching.